Hello friends, I, Mrs. Vaishali Tire from the Department of Microbiology, KTHM College, Nasik, am here to present my topic on major histocompatibility complex in the faculty training program. Major histocompatibility complex is the complex that helps to discriminate between the self and the non-self antigens and to fight against the non-self antigen without acting on the self antigens. It is basically a cluster of genes that are found in all the mammals. Its products help to distinguish between the self antigens and the non-self antigen such that the non-self antigens can be eliminated from the body without causing any harm to the body. The products of this MHC help the immune system in both the humoral as well as the cell-mediated immune responses. These are nothing but the antigen-presenting structures which help to present the processed antigens to the proper immunocompetent cells so that these cells can help in the removal of the antigen from the body. The genes of MHC are located on the short arm of chromosome number 6. And in humans, this complex is also referred to as the HLA complex, that is the human leukocyte antigen complex. The genes of this MHC can be classified into three different classes of genes, namely class 1, class 2 and class 3 genes. The class 1 genes are the genes that encode for glycoproteins that are expressed on the surface of all the nucleated cells in the body. The main function of the products of this class 1 MHC genes is to present the antigen to the TC cells. Mainly the internal antigens like the tumor cells, the virally infected self or altered self cells are the cells that can be eliminated by the activation of the TC cells after they have been presented with the specific antigens. The class 2 genes are the genes that encode for products that are glycoproteins which are expressed on the surface of the antigen presenting cells, mainly the macrophages, the B cells and the dendritic cells. No other cells in the body express the class 2 MHC molecules. The main function of these class 2 MHC molecules is to present the processed antigen to the T helper cells so that the external antigen that has entered into the body can be easily removed by the activation of the T helper cells, which in turn activate the B cells to produce the antibodies and remove the antigen from the body. Class 3 MHC genes are no glycoproteins, or the main function of these genes is not to present the processed antigen to the immunocompetent cells, but the products of the class 3 MHC genes include the secreted proteins that have immune functions, basically the B components of the complement system as well as the tumor necrosis factors and the inflammatory proteins. This is the organization, as you can see, of the HLA complex in the human body. It is class 2 genes are clustered in a region which is DP, DQ and DR region. The class 1 genes are clustered as B in the region of B, C and A, and the gene products of class 2 MHC molecule are the DP alpha beta, DQ alpha beta and the DR alpha beta. While class 1 MHC genes encode for the gene products that is HLA-A, HLA-B and the HLA-C. The class 3 genes are the genes that are not glycoproteins, but they encode for the C4 and the C2 proteins of the complement system, as well as the factor B of the complement system. In addition to this, the products like the tumor necrosis factors alpha and beta are also encoded by the HLA complex. We will be studying about the class 1 MHC molecule. The class 1 MHC molecule is made up of two different molecules or two different subunits, mainly the alpha chain and the beta 2 microglobulin. The alpha chain is a 45 kilodalton chain that is made up of three domains, alpha 1, alpha 2 and alpha 3 domain. The three domains, alpha 1, alpha 2 and alpha 3, are the domains that lie outside the cell. In addition to these three domains, a transmembrane region is also present in the chain, which helps to anchor the alpha chain in the membrane of the cell. This part of the alpha chain is mainly made up of hydrophobic amino acids. And the last portion of the chain that lies inside the cytoplasm is a small cytoplasmic tail which is made up of hydrophilic amino acids. This alpha chain has three regions, as I said, alpha 1, alpha 2 and alpha 3. The distal alpha 1 and the alpha 2 regions are the regions which make up the structure of the peptide binding cleft. The peptide binding cleft, which is made up of the alpha 1 and the alpha 2 domain. The alpha 3 domain is the domain which is very much similar to the second subunit of the MHC molecule, that is the beta 2 microglobulin. This beta 2 microglobulin is a 12 kilodalton protein that is encoded by genes that lie outside the MHC complex. For the expression of this expression of this class 1 MHC molecule on the surface of the cell, it is necessary that the beta 2 microglobulin should be associated with the alpha 3 domain of the alpha chain. Both these components, the alpha 3 domain and the beta 2 microglobulin, show folds which are similar to the folds that are observed in the structure of immunoglobulins, and therefore this particular glycoprotein is said to be a protein that belongs to the immunoglobulin superfamily of proteins. Class 1 MHC molecules are the molecules that usually present the endogenous antigen. That means any antigen that is created inside the body of a particular individual is referred to as an endogenous antigen. For example, if a viral particle enters inside the body and infects the host cell, this virally infected host cell is an endogenous antigen that is produced in the body. Any tumor cells are the cells that are the self cells which have undergone some mutations, and therefore the structure of the cell has changed, and thus it is produced inside the body and so called as the endogenous protein. While the third example of endogenous protein is the altered self cells, which may be altered due to a variety of different regions. So when an antigen is an endogenous antigen, this antigen is processed within the cytoplasm of the host cell, and after processing the antigen is presented with class 1 MHC molecules to the specific TC cells or the cytotoxic T cells, which help to lyse the antigen and get rid of the antigen. We've seen that in the class 1 MHC molecule, the peptide binding cleft is made up of the alpha 1 and the alpha 2 domains. This peptide binding cleft of the alpha 1 and the alpha 2 domain can bind to the small proteins which have around 8 to 10 amino acids, but the most favorable peptide that can be bound in the peptide binding site of the antigen is the peptide that is made up of preferably 9 amino acids. The peculiarity of this peptide that binds in the peptide binding cleft is that at the ends, at the C terminal and the N terminal, it has to have the hydrophobic amino acids such that these hydrophobic amino acids can bind with the amino acids that are present in the peptide binding site. For example, in this figure that is adopted from the book of Immunology by Cube, the 5th edition, it shows that glycine that is present at the second position in this chain and another amino acid present at the 9th position of the chain are hydrophobic in nature. Right? And these amino acids are the ones that bind to the amino acids that are present in the peptide binding cleft, and the binding may lead to the formation of a bulge because of the size of the peptide binding cleft. Thank you.